Good morning, everyone. My name is Elizabeth, and I'm an Associate Director of Product Marketing at PacBio. Today, I'm going to talk to you about full-length RNA sequencing using the Kinnex full-length RNA kit. Last year, PacBio released the Kinnex full-length RNA kit for scalable, cost-effective full-length RNA sequencing. To use the Kinnex full-length RNA kit, first, generate up to 12 different barcoded cDNA using the Iso-seq Express 2.0 kit. These barcoded cDNA can then be pooled for Kinnex concatenation using the Kinnex full-length RNA kit. Additional multiplexing is possible with 4 unique barcoded Kinnex adapters. I'll explain in the later slides how these barcodes work in analysis. From total RNA to generating cDNA using the Iso-seq kit to generating sequencing-ready library using the Kinnex kit takes 2 days. The expected throughput on Sequel II and IIe systems is 15 to 20 million full-length cDNA molecules and 30 to 40 million such reads in Revio. We'll also talk about how the latest SmartLink 13.0 generates isoform classification with abundance information, so you can go directly into tertiary analysis. Different study goals require different sequencing depths. For isoform discovery and comparison of moderate to rare transcripts, such as comparing disease to normal with 2 replicates each, we recommend 4-plex on Revio, which would achieve a target depth of 10 million reads. For isoform discovery and comparison of highly expressed transcripts and cohorts with large number of samples, we recommend a target depth of 5 million reads, enabling an 8-plex on Revio. For plant and animal genome annotation, we recommend either multiplexing 4 tissues or 8 tissues of the same species to run with Kinnex on Revio. We demonstrate that the Kinnex full-length RNA kit increases throughput without biasing transcript capture. We took the same UHRR universal human reference RNA sample and sequenced it both with Sequel II and Revio with and without Kinnex. We achieved 3.2 million reads without Kinnex on Sequel II. This was increased by 6-fold to 20.5 million reads with Kinnex on Sequel II, and a 16-fold increase to 47.3 million reads with Kinnex on Revio. Further, in comparing the transcript length distribution, we show that there is no bias in transcripts captured when using Kinnex concatenation. Comparing the abundances of the known isoforms also showed that Kinnex concatenation does not skew isoform abundances. We've applied Kinnex full-length RNA kit to a variety of tissues and species. The minor transcript length distribution, as we observe here, are largely due to RNA quality, tissue, and species difference. This slide demonstrates why we recommend 10 million reads per sample. Over a variety of samples, we observed, using subsampling analysis, that around 10 million reads, 80% of the known isoforms are detected. We then did a simulated study where we took the WTC11 sample, subsampled it to 10 million reads, creating a 4-plex run, and showed that on average, each sample would detect 17,000 genes or 47,000 isoforms. Using the US list price for Kinnex and Iso-seq kits as well as SMRT cells, this would come to $488 cost per sample from with an all-in cost of including cDNA generation, Kinnex library concatenation, and SMRT cell sequencing. If we reduce the sequencing depth to 5 million reads per sample, allowing for an 8-plex for Kinnex on Revio, we would achieve 16,000 genes, 42,000 isoforms with an average cost of $304 per sample. On Sequel 2, we recommend no more than 3-plex with a target depth of 5 million reads per sample. This would capture 50,000 genes and 39,000 isoforms. Based on the US list price, the cost per sample would be $698. However, we have an ongoing promotion until the end of March, where for the first Kinnex kit purchase for Sequel 2 sequencing, you get every SMRT cell for free for each of the 12 reactions. Let's now talk about bioinformatic analysis for the Kinnex full-length RNA kit starting SMRT-Link 13.0. We've talked about how barcodes are incorporated into the molecules during the library preparation. The Iso-seq Express 2.0 kit incorporates up to 12 unique barcoded cDNA primers. The concatenation of these cDNA are random. After the array is formed, 4 unique Kinnex barcoded adapters can be used. Note, however, that the Iso-seq analysis workflow, which I will show in the subsequent slides, only consider the barcoded cDNA primers for differentiating different samples. The order of bioinformatic analysis is thus: first, demultiplex the Kinnex barcoded adapters. This could be done on or off instrument. Read segmentation to deconcatenate the reads, and Iso-seq for full-length isoform analysis. In the next few slides, I will walk you through each of these steps for analysis, including deconcatenation, primer extraction, clustering, mapping, and classification. First, HiFi reads are deconcatenated to represent its original full-length cDNA molecules. This is done at the command line using Scera. The next step, the Lima tool, removes barcoded Iso-seq primers. Iso-seq Refine further removes the poly A tails and any remaining artificial concatenamers. We then perform de novo isoform clustering on the full-length reads. The tool to do this is Iso-seq Cluster Tool, a new version of isoform level clustering scalable to Kinnex throughput. The previous Iso-seq cluster tools are not scalable to Kinnex throughput and is no longer recommended. The new algorithm follows a coarse to refined clustering scheme, where different reads are already first roughly clustered based on their k-mer similarities. Then, using sequence alignment and nearest neighbor alignment, these subsequent coarse clusters are further refined. The algorithm does not load the entire dataset into memory, making it scalable to any size. We now recommend Iso-seq Cluster 2 for any PacBio Iso-seq data clustering. After generating isoform level consensus sequences, we then use Minimap2. The wrapper is called PBMM2 to align them to the reference genome. We then further collapse any redundancies based on the exonic structures using Iso-seq collapse. After this step, we now have a mapped set of unique isoforms. Pigeon is the tool to classify isoforms into different structural categories based on a reference annotation such as GenCode. Pigeon is the PacBio implementation of SCONTE3. There are a few major categories of isoform classification. Full splice match, or FSM, means all the splice sites from an Iso-seq transcript matches the reference. In other words, it's a known transcript. ISM, or incomplete splice match, means it matches the reference for every exon but is missing some of the first or last exons. This could be alternative start sites, end sites, or 5' degradation. There are 2 categories of novel isoforms. NIC, novel and catalog, are novel isoforms using a combination of known splice sites. Novel not in catalog, or ANC, are novel isoforms with at least one new splice site. Other categories such as genic intron, genic genomic, or antisense are quite self-explanatory. Pigeon also has a step called filtering that removes any potential library artifacts. Most of the time, this removes reads that are intragenic, genic intronic, or genic genomic. There are a few other auxiliary tools in Pigeon that might be helpful, including Pigeon Report, which subsamples reads to generate gene saturation or isoform saturation curves. Note that SmartLink only supports a preloaded version of human and mouse genome and annotation. If you want to use a different GenCo annotation that's in SmartLink, or you are not working on human or mouse, you need to prepare a separate set of custom reference genome and annotations. This can only be run on the command line. Pigeon Prepare will help you make sure that these files are the correct format. Here are some of the screenshots of running SmartLink 13 for read segmentation and Iso-seq. You can see this live on a recorded video on PacBio's YouTube channel. Just go to the Kinnex playlist. Here's an example of the report for transcript classification. You see that the categories from the different pigeon classifications that I've just explained to you are shown in this table. SmartLink 13 enables analyzing Kinnex full-length RNA data both separately and together for barcoded cDNA. In this report, each barcoded cDNA is analyzed separately, so you see different pigeon reports for them. This concludes my talk. Thank you for listening.
Hi, everyone. My name is David, and I'm a PhD student at the University of Zurich. Today, I'm pleased to give this webinar on transcript detection and quantification using Kinnex full-length RNA sequencing data. First, I'd like to note that this is joint work with Madison Malferber at the University of Virginia, which runs supervision by Gloria Schenkman at the University of Virginia and Mark Robinson at the University of Zurich. To kick us off, I'd like to pose the big picture question that we'll be mostly talking about today, which is which of the three major RNA-Seq application groups does Kinnex RNA-Seq data enable today? And we broadly define these three groups as pure quantification based, also downstream quantification based tasks, which we exemplarily show here via differential transcript expression. But this also applies to tasks such as differential transcript usage or differential gene expression. And the third group are isoform discovery tasks, which are generally called transcript assembly for short read RNA-Seq. And I want to note briefly why we're doing this, or specifically how this relates to previous work, because we are standing on the shoulders of giants, so to speak here. So there's many papers, obviously, but the two main ones that I'd like to mention are lrGasp, which is the first one here, which benchmarked Oxford Nanopore, Illumina, and PacBio together actually for quantification and also other tasks such as isoform discovery. And also a recent work which compared Illumina to Oxford Nanopore on these three groups. The key thing in both of these studies, well, in lrGasp, PacBio data was present, but it was much lower yield since it was pre-Kinnex. And in the second paper here from Nature Methods, PacBio was also not included, also due to pre-Kinnex yield limitations. And so today, we're really interested in exploring some of these, or all of these three groups, and specifically for Kinnex data, right, which enables high yield RNA-Seq data for PacBio comparable in terms of yield to Illumina. So before I dive into our own data set, I want to briefly talk about lrGasp and how it really motivates a lot of our work. So lrGasp is a very large project that had multiple challenges, and I'll very briefly talk about the challenge setup in a second. But before that, I just want to say that lrGasp really had a lot of different data sets. So there are different species, sample types, protocols, and then finally sequencing platforms, so including PacBio, Nanopore, and Illumina, as I mentioned. And in lrGasp, what you actually saw is that cDNA PacBio, again, this is pre-Kinnex, had the highest read length, which you see in this plot here, and also quality, which you see in this plot here, but it did have the lowest yield, as you can see here, in terms of read numbers in millions. And this is because this is pre-Kinnex. And now post-Kinnex, or with Kinnex, this is really no longer the case, because we can maintain these high read length and qualities, as Liz has shown, but we can get much improved throughput. Also, as I already mentioned, lrGasp has the three types of challenges. The second is form qualification, of which you can see some of the results ordered by tools and metrics, as well as data sets up here. I'd like to note that IsoQuant and Bamboo were among the top performers in lrGasp for quantification specifically, and because of that, combined with the fact that they're very popular in the open source community, we will all, along with another tool called ORFISH today. Moving on to our dataset. Our dataset encompasses an iPSC to EC differentiation. iPSC stands for induced pluripotent stem cells, which is our day 0, and EC stands for endothelial cells, which corresponds to our day 5. There is a time course along this differentiation, but since our work today is really focused on this classical 2-condition setting, I won't really talk too much about the time course. Before I talk more about the dataset, I just want to note that all this experimental work, as well as the figure, were done by Madison from the Shenkman lab at the University of Virginia. The number of files here on both day 0 and day 5 is actually representative of the number of samples. So we have 3 replicates on day 0, as well as 3 replicates on day 5. After that, we do standard library preparation, followed by Kinnex concatenation and sequencing on the Revio for PacBio, and standard library preparation, followed by Illumina sequencing for the short reads. In addition, we also sequence our dataset with surf spike-ins for additional QC and ground truth. These surfs are made by Lexigent and are essentially synthetic RNA controls that consist of a total of 69 artificially constructed isoforms that range in length from 191 base pairs to 2.5 kb. These are specifically designed not to map to any human or mouse genome, and such that you can easily separate them from real data and thus use them for quality control and other things. The surfs consist of 4 submixes. So each surf mix has 4 submixes. Apologies, the terminology is a little bit confusing, but you see the submixes here in these 4 different colors, and you have the mixes that are indexed by E0, E1, E2. So the mixes are just what you spike in. So in particular, in our example, we spiked in the mix E2 into day 5 and the mix E1 into day 0. Since we know the relative quantities for each submix in each mix, so in particular for E1 and E2 here, we can also calculate the expected fold changes for each submix when we compare E2 over E1, which is exactly the comparison we have, right? So if we compare a replicate of day 5 to a replicate of day 0, the fold changes that you see here are the expected fold changes. So for example, for submix 3 that you see down here, we expect a fold change of 1 over 32, whereas for submix 4, we expect no change because it's a fold change of 1. Okay. Moving on. This is an example of what the serves actually look like in IGV. And I just want to emphasize that the serves are not necessarily the most biologically realistic if you're used to, let's say, real transcript, especially some of the longer ones that you're perhaps interested in capturing with long reads. In particular, as I already mentioned, the serves can be quite short, right? So the longest serves, there is actually a separate long serve set, but in our setting, the longest serve is 2.5 kb, which is not particularly long. And there are a lot of these monoexonic serves, as you see here with serve 206 and 205. In addition, you have a lot of serves with very minor differences, right? So you see, for example, serve 201 and serve 202 here, which actually, it's a bit hard to see here in the track, but they have very minor differences at both the 5 and the 3' end on the order of, I believe, 5 to 20 bases, which can, make quantification and other tasks really difficult, right? Because these 2 isoforms are almost the same, and probably to a degree that you might not see in real data regularly. Okay, moving on to some general preliminaries. So throughout the webinar, I will liberally switch between serve and quote unquote real data results. So when I say real data, I just mean data from human on GenCode from the cell line that we introduced. So since we do not have ground truth for expected or differential expression, I will be using the serves for benchmarking these aspects. At the same time, certain statistics will be much more interesting for you on real data. For example, the number of genes or isoforms detected, as well as differential expression on a biological question of interest. Because of that, as already mentioned, I will switch liberally back and forth. The slide title will make very clear whether I'm talking about serve or GenCode data. In addition, I just wanted to emphasize one more time that the serve data is not necessarily completely biologically realistic. So maybe take some of the serve results with a little bit of grain of salt, because it's perhaps not perfectly characteristic of the type of data that you're interested in. Some more general preliminaries or caveats rather, before I really get into some of the methods and some of the results. This is not meant to be an exhaustive benchmark. meaning we chose a few representative tools, but we don't claim to be doing a complete exhaustive benchmark here. In addition, a lot of the tools, especially the long read tools, are still very much in flux, so this is a snapshot as of today. In addition, I'm not making any blanket statements here about technology A or method A is good or bad. This is really just a very specific setting, right, with these specific tools on these specific data sets. Lastly, we're focusing on state-of-the-art tools according to previous benchmarks. In particular, that means we did not include the Iso-seq workflow that Liz showed you earlier because it was not benchmarked in lrGASP. So we're focusing on other tools here. Generally speaking, the goal of this webinar is to show you some of the things that you can do with Kinnex RNA-Seq today and how to do them. Okay, getting a little bit closer towards quantification. One other thing I wanted to briefly highlight is that quantification is actually the hard part of this pipeline, right? So suppose you have some FASTQ files that contain Illumina reads and some other FASTQ files that contain Kinnex reads. Obviously, the length of the reads here is not to scale. Getting towards this count matrix is where you might actually find quantification differences between Illumina and Kinnex, right? Because perhaps you want to model different biases. For Illumina, you maybe want to model the fragment length, whereas that's probably not necessary for Kinnex because there are no fragments. Generally speaking, you have full-length reads. So that's why you have these two different arrows. So you do perhaps different quantification methods and you get to two count matrices. Once you have this count table, the downstream tools are actually the same because the downstream tools don't care where this count matrix comes from, right? They only care that this count matrix is accurate. So as long as your count matrix is accurate, which is maybe a big if, right? But if that is the case, then the downstream tools, such as differential transcript expression, differential gene expression, differential transcript usage, all the same. And you can use the same tools that have been developed for several years already for short read RNA-Seq data. There's a very small asterisk here regarding things like quantification uncertainty, which some quantification tools can provide and which you can actually provide to some of these downstream tools. But yeah, I will leave that aside for today. Okay. Talking about tools. So our goal, like I said, was to consider some representative tools for both Illumina and Kinnex. So really to simulate a little bit the setting that you are probably in, namely that you sequence Kinnex RNA-Seq today, and you're interested in analyzing it, what would be maybe some of the typical methods that people would use. So for Illumina, we use Scallop for isoform discovery / assembly, Salmon for quantification, and for both Illumina and Kinnex, we use EdgeR for DTE and DGE. And apologies, maybe I didn't clarify those abbreviations before, but DTE refers to differential transcript expression and DGE refers to differential gene expression. For Kinnex, we use Bamboo for isoform discovery. And for for all of the real data results, we use Bamboo for the quantification. And for the serve quantifications, we additionally add IsoQuant and ORFISH to get some sense of comparison between the three tools. I will also note that ORFISH was really just made available, so hence why I'm only citing the GitHub repo and not a paper or a preprint. Okay, moving on to the quantification tools. So what I want to highlight here is some of the different variants that you might encounter when you're doing quantification specifically for Kinnex RNA-Seq. So some of these are the type of alignments used, the type of quantification used, as well as the language and compute requirements. For the type of alignment, the tools we're using differ between transcriptome and genome. So Bamboo and IsoQuant use genome alignments, while ORFISH, similar to Salmon, uses transcriptome alignments. For the type of quantification, both ORFISH and Bamboo use a variant or just the traditional expectation maximization algorithm. I should also note that ORFISH also has additional filtering options and a coverage model which is designed to improve quantification accuracy. IsoQuant opts for a more traditional read splitting approach as well as additional filters. Bamboo is written in Rust, while IsoQuant is written in Python, and Bamboo is written in R, IsoQuant is written in Python, and ORFISH is written in Rust. We did some small, again, this is not a complete benchmark, but we did some just small preliminary computational experiments, so just quantifying 1 million reads, serve reads in this case, for all of the tools, repeating this a few times, and looking at both memory and runtime. And we see that ORFISH is really by far the best in terms of both memory and runtime. Yeah, so take that as you will. Moving on, I wanted to give you a small example of a typical open source workflow, similar to what Liz showed you for Iso-seq. This is some somewhat simplified code. I mean, most of this should run out of the box, but sometimes I simplified it slightly just for ease of exposition and to fit things on the slide. So since PacBio provides unmapped reads in UBAMs and not in FASTQs, the first step would typically be a conversion from UBAM to FASTQ. So you can use PacBio specific tools here, or SAM tools can typically also do this. Following that, you want to map your FASTQ to either the genome or the transcriptome. Generally speaking, most people really use Minimap today, and the PacBio wrapper around Minimap PBMM2 also uses Minimap under the hood. And yeah, you might want to pay attention to some of the parameters depending on what you're mapping to, genome or transcriptome, but the documentation is really very good and yeah, should clear everything up. Following the mapping, we have the isoform discovery step, which for a lot of methods, such as Bamboo, as well as IsoQuant, actually often includes quantification. So here's an example invocation for Bamboo. And you can see, I mean, this makes it very easy for exposition, but basically Bamboo specifically has parameters that say discovery equals true, quant equals true, which means, okay, if you pass both as true, it does both isoform discovery and then directly does quantification on this novel annotation. And yeah, so this is quantification against genome mappings. Alternatively, you can also do transcriptome-based quantification using, for example, ORFISH, as I introduced earlier. In this case, from the isoform discovery step, you would go back to the mapping step because you would extract your potentially partially novel transcriptome and map against this, and then pass that into ORFISH. Finally, you would use, or you could use, if you're interested in this type of thing, a differential expression. Here's again, somewhat of a outcut of a typical EdgeR workflow, where in the end you can look at the top tags, or also just let the test run this decide tests function, which essentially just gives you a vector saying upregulated, downregulated, no change. So especially EdgeR really has a lot of different options, so I highly encourage you to read the manual and look at the vignette to get some more detailed ideas of what you can do downstream. Okay, moving on to the results section. Before I come to the actual topic of this webinar, namely quantification, as well as downstream tasks such as DTE and DGE, I want to briefly highlight isoform discovery again, because this is probably one of the applications that made a lot of you want to try long reads in the first place. And so what we did here is we performed isoform discovery completely de novo on the serves. So essentially we're rediscovering the serve annotation, and we see, I think, a relatively typical picture, namely that long read-based RNA-Seq data, in this case Kinnex, really strongly outperforms Illumina, especially in terms of precision, right? So Illumina suffers from a lot of false positives in typical assembly tasks, whereas the recall is also a decent amount higher for Kinnex, but it's really the precision that's yeah, like 3-4x what it is for Illumina. So yeah, I think coming back to this first task of isoform discovery, right? I think it's clear that in our particular setting, really Kinnex really strongly outperforms Illumina in terms of performance, in terms of isoform discovery. So having given that, I want to move on now to quantification and really talk about, okay, Kinnex can do really well compared to Illumina, or really better than Illumina at isoform discovery. What about quantification? But before we really look at the quantification results, let's just make sure that we're actually comparing apples and oranges in terms of read numbers, right? I told you that Kinnex, and also Liz told you that Kinnex really has a strongly increased throughput. So what does it actually look like relative to Illumina? And we look at the number of total reads on our samples. So we see that basically all of our samples have at least 40 million reads, and then depending on the technology and the day or the rep, sometimes we have a little bit more, a little bit less. But generally speaking, these numbers are fairly comparable. I mean, Illumina sometimes has a little bit more, but also the case for Kinnex. When we look at total base pairs, we see something perhaps unsurprising, right? That Kinnex has obviously much higher total base pairs for equivalent number of read depths, which is because Kinnex obviously has much longer read length, right? So Illumina in this case is paired end 150 base pairs, and Kinnex has a read length distribution that looks very similar to Iso-seq, where you get medians on the order of a few kb, which is much longer than Illumina, and thus leads to much higher number of total base pairs. Okay, getting to the meat and potatoes of the webinar. let's talk about actual quantification. So one of the first things that people often ask themselves when you're talking about quantification is how replicable is it? So when you do multiple replicates, how close are they to each other? And so this is really what we're looking at here. So I'll walk you through this plot because the heat map is a little bit confusing at first, perhaps. So in green, we have Illumina, in pink, we have Kinnex, in blue, we have day 0, and in orange, we have day 5. So the first thing I want to look at is basically the quadrants of the diagonal, because they tell us how replicable is each technology within technology, right? So this quadrant is day 0 for Illumina, day 5 for Illumina, day 0 for Kinnex, day 5 for Kinnex. And I think you can see quite quickly that these 4 quadrants look very similar, right? So, I mean, I don't want you to focus too much on each exact number, right? The difference between 0.88 and 0.94 or something is not really significant here or relevant here. But generally speaking, what we see is that Kinnex quantifications really show within technology and within day show very competitive replicability to Illumina, really kind of no difference in terms of replicability here. You can also compare between day because we did perform a differentiation, but that doesn't affect every single transcript, right? And so we see for both Illumina and Kinnex also relatively high correlations between days. So this is essentially the comparison here or here. And we see actually that Kinnex has slightly higher correlations, but again, this is a few percentage points, so it's nothing I would really try to interpret. And the other thing that's interesting to look at is between technology comparison. So what happens if we compare Illumina on day 0 to Kinnex on day 0? And here we see correlations on the order of 0.7 to 0.75. And again, this is relatively reassuring because, yeah, I mean, this is real data. It's on the transcript level. So correlations of 0.7 are actually already, I would say, quite good and really show that not only is Kinnex or does Kinnex seem to be replicable, it also achieves, I would say, decent to good concordance with Illumina data, which yeah, is very good and very reassuring. You can look at similar plots, scatter plots essentially, where you can do this on the gene level as well as the transcript level. And here we just get some kind of additional information, right? Because we can really look at some of these boundaries, for example. And for example, here, you can see some things that are maybe expressed in one sample, but have very little expression in another sample, something like this. But I would say for both Illumina and Kinnex, yeah, we see, I think, kind of what we want, right? That you see basically everything following the diagonal here. You see maybe for Kinnex here, you see a few more things really off diagonal here, something like this, where you see things that have actually moderate expression in one of the samples and very low in one of the others. It's generally speaking, not what we want, but we also do this a little bit in Illumina, right? So I think it's nothing to worry about. I should also mention that the numbers here are lower for Illumina, but this actually has a reason, which I will get to in a second. Namely that Illumina actually detects more transcripts and genes, which means that these numbers aren't exactly comparable, right? Because for each technology, I only look at things that are expressed, and since not the exactly same things are expressed for Illumina and Kinnex, you can't exactly compare these numbers, whereas here you could. Okay, here we're talking about the detection as I promised. So long story short, Kinnex really detects a very similar number of genes, but it does detect fewer transcripts than Illumina. So when you look at the number of genes detected at both full depth and downsampled up to 5 million reads, we generally see that Kinnex detects about 200 to 300 less genes compared to Illumina, whereas for transcripts, it does detect a much larger number of fewer transcripts. So at full depth, I believe it's around 10,000 fewer transcripts, but it does shrink quite quickly. And at 5 million reads, it's down to about 1,000. Yes. Okay, moving on to downstream tasks. And I think this is where it really starts getting very interesting. So the first thing we actually did on real data was saying, okay, we don't really have ground truth here, but what we do have is we know that we performed this differentiation from these induced pluripotent stem cells towards endothelial cells. So how about we take some endothelial cell marker genes from a typical single cell database, in this case Panglao, and we actually look at these markers and we check which of them is upregulated in Kinnex as well as Illumina on day 5 compared to day 0. First thing I should note is that we find 75 here that are actually not upregulated in either Illumina or Kinnex. This is a bit of technical reason because we didn't filter the marker genes here for the type of differentiation that we're doing, but actually not all the marker genes here are really relevant for our case. But the ones that are found by either technology, we actually see that almost all of them are shared, right? So we see 102 marker genes that are found by both Illumina and Kinnex to be upregulated, and only 6 and 4 respectively that are only found by one of the technologies, which again, I think is very high consistency and really gives us confidence in the Kinnex quantifications, because for the things that really matter, for example, differential expression, they really produce results that are very, very close to Illumina. I should note that the type of differential expression here is quite strong, right? So it's really a differentiation. So in some biological conditions, you might not see differential expression that strong, but nevertheless, I think it's very, very reassuring regarding the Kinnex quantifications. Okay, moving on to the surf quantifications. So I'll walk you through this plot because it's a little bit much to take in the beginning. So we have surf length on the x-axis because we're maybe interested in if some of the technologies have a length bias or not. On the y-axis, we have observed log-fold change based on each technology. And then recall that we have ground truth data from the surfs that I showed in the very beginning, right? Because we have the relative quantities for each mix, and so we can calculate the expected log-fold changes. So the expected or theoretical log-fold changes are in these colors here. So blue corresponds to -4, and so on. I've also added these guiding lines in the respective color as well. So what we want to see is a tight clustering of colors around the line of the same color. So in particular, for example, here, would really want to see something like this, where we see the points, blue points, clustered very tightly around the blue dashed line. And as I promised, here we're showing Illumina data, which is salmon quantifications, as well as 3 different Kinnex methods, because here it actually becomes very relevant which quantification tool you're using. So let me talk about Illumina first. So Illumina, generally speaking, shows very tight quantification. So most of the time we see exactly what we want to see, right? So we see, generally speaking, quite tight clustering. We do see a few outliers, like here, for example. We also see some slight flips, for example, here, where the orange points, which are actually 0, are closer to 1 than the 1 points. But yeah, stuff like this, I think, is still to be expected. When we look at the Kinnex quantification, I will first make some general comments about the Kinnex quantifications and then go tool by tool. So generally speaking, for a lot of the Kinnex quantifications, they also look actually very good. So for example, if you look at bamboo here and you look at these pink points here, green points here, orange points mostly here, it all looks quite good. And this is consistent between tools, that I would say most of the points are quite good. And there's 2 things I think now to point out for Kinnex. So one thing is that it does struggle for some of the quantification tools for things that are very short. So things below, say, 500 nucleotides. So there would be line, let's say, here. So you see some of these points here. I should also emphasize that for oarfish, this has a little bit to do with the filter, because oarfish does have some filtering options which take out certain alignments which are deemed to be untrustworthy, or perhaps not to be advantageous for the quantification. But it will actually show that this can be these filters can actually really help. So that's the first thing to point out for Kinnex. The second thing to point out for Kinnex is that you get some of these outliers. So for example, here's one for isoquant, right? So this should have a log-fold change of 0, but it actually has a log-fold change of -11 or -12. And for most of the Kinnex tools, you can find some of these points. And some of you might have already noticed, right? Something that's a little bit weird with this plot. Namely that the outliers aren't really consistent between tools. And here's an example for isoquant, right? We have this point here, but actually neither for oarfish nor for bamboo, we really see this point. And that's something I want to highlight. So this is actually surf 303. So when we actually look at the counts, so this is just the counts for day 0 for the first 3 reps for surf 303. So we see that it has 0 counts on the first rep, 0 counts on the second rep, and actually above 200,000 counts for the third rep. And when we look at the tracks for this, we see that this is actually an isoform that is fully contained in another isoform. So 303 is fully contained in surf 301, right? And when you actually dig into the code, isoquant actually has a line that says roughly, I'm only going to give any counts or any quantifications to features, in this case isoforms, that have a unique splice match. And unique really here means that it's roughly speaking a fuzzy perfect match. So in isoquant lingo, surf303 could still have a perfect match, right? Like if you have a read that matches exactly surf303, it would be considered unique to surf303 and not also a partial match to 301. And so what happens is actually that in day 0 rep 1 and day 0 rep 2, you actually don't get a perfect match to surf303 at all. You only get matches that are ambiguous with surf301 because they don't match the coordinates of surf303 perfectly, right? And again, there's some fuzziness, but it's for PacBio, it's not super fuzzy because generally speaking, PacBio is quite accurate. But if you get truncations, for example, isoquant in that case can no longer really tell if this is unique. It's not considered a unique read for either 301 or 303. And so even though there are a lot of reads that could be potentially matches for surf303, in this case, isoquant says, since it's not a unique splice match, I don't think it's a real feature, and I'm going to give it 0 counts. And I'm not saying this or showing this to really put isoquant down or anything, right? I think, again, the goal isn't really here to compare things, but I think upon first glance, isoquant actually looks the best here, and this is consistent with lrGASP. The point is more that, especially for things that can be a little bit weird biologically, like the surfs, you should really look at your quantification carefully and really, if you see something like this, really think, okay, why could this be happening? And it's always anyway a good idea to dig into the alignments, but you should also be aware of the fact that it can also be something tool specific, right? Especially if this is not consistent across different tools. Okay, getting to the last points, differential transcript expression. So this is again on the serves. So here we look at each of the quantifications and look at the number of the true positive rate as well as the false discovery rate. And so we see first thing that Illumina does perform slightly better, I would say, than the Kinnex-based tools. But we also see that actually the Kinnex-based tools do show lower FDR, with ORFISH actually achieving 0 false positives, which is what I mentioned earlier with the filters, right? So this is really helping ORFISH, the filters here, and otherwise it really has 0 false positives. And I should also note that outliers like the green ones here actually don't lead to any problems, right? Because they're in the right direction, quote unquote, because these green points, they should have a log fold change of 1, and they actually have an observed log fold change of whatever, 10 or 12 here, but it's still in the right direction, because if you actually have to decide, that still counts as an upregulation. It's just an upregulation is too strong, right? But if you take this as a binary, it's still upregulated, which is correct. And yeah, so GTE, I would say also for Kinnex looks actually quite good, right? So we do see slightly lower TPR than Illumina, but we also see lower FDR, and especially considering some of the things that I said about this serve weirdness combined with the tools, I think these GTE results for Kinnex also actually look very good. Okay, so getting to the conclusion. So there are already various tools available for all parts of this Kinnex RNA-Seq pipeline today, and there's more being created every day. Here's an incomplete list of tools, and I encourage you to try all of these out. I've used all of these myself, and I think they're all quite easy to use and very useful. And perhaps they can supplement some of your workflows with Iso-seq. Okay, so coming to the overall conclusion, the question of which of these groups of tasks Kinnex RNA-Seq can already enable today. So I think, I mean, I showed this in the beginning, but also I guess most of you are aware of this, that Kinnex RNA-Seq or PacBio long-read RNA-Seq in general performs extremely well for isoform discovery. The question that we're interested in today is how well does it perform for quantification and these downstream tasks like differential transcript expression? And I would say actually quite well. So for the quantification, we did see some of these outliers, right? But generally speaking, the quantification was already very concordant with what we would expect. And I already showed as well that a lot of these outliers can actually be tool specific and are often related to the sort of weirdness of the serves, which means that often it actually is not necessarily a data problem, but can actually be fixed on the method side potentially, or perhaps not fixed, but really accounted for, right? And also for downstream tasks such as differential transcript expression. So we saw very high concordance between the endothelial cell markers that were upregulated between Illumina and Kinnex, and also on the serves for the DTE, we saw TPR that was perhaps slightly lower than Illumina, but still reasonably high, I would say. And at the same time, we did see lower FDR, which is very encouraging. And yeah, I think with that, Kinnex RNA-Seq really can already start enabling all of these groups today. And yeah, I think that things will only improve as time goes by, right? Because obviously both the sequencing and the methods are currently constantly being improved and worked on. And yeah, I think with that, I'd like to conclude and just thank my lab at the University of Zurich for helpful discussions and feedback, and note that this work was supported by an SNF grant. And lastly, just say that we're very grateful to PacBio, especially Liz and Roger, for the collaboration and sequencing this cool data set for us. And with that, thanks a lot for your attention. I hope I got some of you excited about Kinnex today. And with that, I'm happy to take any questions.
Thank you for listening to the recording. So please use the questions chat box to type in any of your questions. We already have some of these, so I'm going to start answering. Me and David are going to start answering them. So first, I'll answer some of the high-level questions. People asked, what is the difference between the terms Iso-seq, MAS-Seq, and Kinnex? So actually, in terms of like chronology, Iso-seq came before MAS-Seq and before Kinnex. So Iso-seq was used to both describe the library preparation of full-length RNA sequencing with PacBio and also the bioinformatics. So you'll see a lot of the bioinformatics tools start with like Iso-seq cluster, Iso-seq workflow. This is before the Kinnex/MAS-Seq concatenation came along. So the original concatenation method for increasing throughput by concatenating these small amplicons, which both includes the cDNA from bulk that you're hearing today, but also single cell like 10x cDNA, that method was called MAS-Seq, and that term came out around 3 years ago. We had a kit that was released 2 years ago called the MAS-Seq for 10x single cell 3' kit. That was the first to utilize this concatenation method, but for single cell only. And what we released last year were 3 Kinnex kits. The Kinnex full-length RNA kit is the kit we're talking about today, that's combined with the Iso-seq Express 2.0 kit for bulk RNA sequencing. So if you're interested in single cell, there's the Kinnex single cell RNA kit. And you can actually see all of these at, if you go to pacv.com/Kinnex, it actually explains to you what Kinnex means and what kits are out there. I'll also answer, I think, another question on Pigeon, since that was part of my talk. So for the Pigeon tool, there are it removes artifacts. So like what's decided as an artifact. So Pigeon is based on the tool called Scanti, SQANTI. I recommend you go check out the original Scanti paper that was published, I think, almost 8 or 9 years ago, but also Scanti 3, which is currently in preprint format, but should be published pretty soon. Scanti actually describes the library artifacts such as intrapriming and RT switching artifacts. They are pervasive in any cDNA generation. Doesn't matter if it's single cell or bulk. And so Scanti and Pigeon actually is able to use some molecular sequencing characteristics to filter these artifacts. Okay, I'm going to start asking some questions for David. So what steps are required for the conversion? So one of your slides, you talked about like, okay, you need to pre-convert something from like BAM to FASTQ. What actually goes into that BAM to FASTQ step?
Right, so what goes in there is essentially the UBAM, so the unmapped BAM file of FLNCs. So this is essentially the output of the third step when you follow the classical Iso-seq workflow. After the first step, you will have a UBAM of FLNCs, and this is what you would then convert to a FASTQ and then map with Minimap and so on. And I will just quickly put the workflow of Iso-seq in the chat as well, so you can know what I'm talking about for steps 1 to 3.
Yep. And then I think also another question is a little more generic on the Illumina. There's actually 2-part question. The first is like, what was the read length that was done with Illumina? I think I can actually help answer that question. Is that it's 100 base pair paired end reads. David, do you know the platform it was done on for the Illumina?
Yeah, so sorry, just a small correction. So it was 150 paired end, but I unfortunately also don't know the platform.
Okay. And then a related question is that in the comparison of Kinnex to Illumina, we showed that they had comparable read depth, read being a loaded word, right? Because the Kinnex reads are much longer. And then later you show that Illumina detected slightly more genes and transcripts. And, do you have some idea about why that's the case?
Right. Yeah. So I mean, assuming that we have roughly comparable total counts, right, which is the case here, sort of just going by elementary algebra, there must be something happening that one of the technologies has more highly expressed and one has more lowly expressed genes, right? So that is what's happening. We're still investigating a little bit more in detail why that could be, right? So one possible explanation would be that perhaps Illumina has slightly higher dynamic range in the sense that Illumina starts detecting things like very lowly expressed things more easily. That's, for example, one explanation. So you have equal total count, but then maybe for something really highly expressed, you have 100 less counts, and then you get 10 things that only have, let's say, 10 counts.
So here's another, I think, good technical question on Illumina, right? So short reads might have more mapping uncertainty, especially for, let's say, homologs or repetitive genes. Did you see any of that manifesting in the comparison?
Yeah, that's a very good question. I mean, I agree that that's the case. I think, broadly speaking, in a lot of the benchmarking things here, these were relatively easy things, like easy in terms of mapping uncertainty doesn't play a huge role there, in particular for the serves, for example, because these are quite short and thus maybe playing more to Illumina's strengths. The other examples that we saw, for example, these ECs, where we had this DGE experiment, I also don't think that mapping uncertainty plays a big role here, but I do think that for real biological applications, if you're interested in, say, alternative splicing, there I think the long reads can make a difference, right? And like I said, I think this will probably come a little bit with time as well, as maybe some of the quantification models get adapted to perhaps some of the stuff like truncated reads and so on. There's some methods already that are trying to model this kind of thing. I think as that progresses forward, we'll see increases in performance, especially in these real biological alternative splicing things.
Cool. And then maybe let me continue the Illumina versus Kinnex before jumping to the Kinnex related tool question. So you actually had two slides where you first do de novo discovery comparison, right? It was before you show quantification, and you show, like, I think bamboo versus salmon. And I believe the message from that slide was that did Kinnex outperform Illumina for transcript discovery, but then later you showed that Illumina detected more transcripts. I think some people were not quite clear how those two slides work together.
Right. Yeah, very good question. So, so conceptually, the difference just here is between sort of detecting known transcripts. So this is where Illumina had higher numbers, right? So this comes a little bit back to this dynamic range that I just mentioned. So if you have a known reference, in this case GenCode, and you quantify against this, then Illumina detected more transcripts at a given read count or CPM threshold. And the discovery is really, okay, either you do de novo, or maybe your annotation is missing some transcripts, then basically how many of the true transcripts does each of the technologies recover, and there Kinnex strongly outperformed, yes.
And this is actually a question coming from me on de novo discovery, is that can ORFISH or IsoQuant be used for novel isoform discovery? Because I know you'd use mostly bamboo in Kinnex.
Yes, so ORFISH is only quantification, only a quantification tool. IsoQuant also does isoform discovery, yes. I would say in general, most of the tools that I listed for quantification, except ORFISH and salmon, basically all do isoform discovery as well. Basically all of the, or well, I shouldn't say all, but 90% of the long read tools that are genome-based that I've seen do both isoform discovery and quantification.
Cool. And then now that you've showcased, bamboo, IsoQuant, and ORFISH, what would your advice for, like, how do you choose between these tools, and should all of them be tried?
Yeah, I mean, that's obviously a difficult question, and I think, like, until we have more complete and rigorous benchmarks, it's something that I can't conclusively answer. So yes, I would say try everything if you have the option. I mean, also keep in mind that the slide I had very early on regarding computational requirements, right? So ORFISH there really strongly outperformed. So it also depends a little bit what you're doing, right? Because if you're sort of just getting started and maybe aren't running on a cluster or something, computational requirements can also play a big role. But yeah, I mean, if you're unsure, then I would definitely try multiple things, yes.
Cool. I think I'll answer this question. It's more on the technology itself. So, someone asked, is cDNA concatenation done on different tissues of the same species only, or can it be done on different cDNA for different species? So The answer is theoretically, there is no reason you cannot mix cDNA from different species for a Kinnex library generation. I think the only concern would be that, we've only tested this on cDNAs that have very similar length distributions, and we knew that it wouldn't cause a bias given how the Kinnex concatenation is currently working. I would recommend, proceeding with caution, that if you're using wildly different species, they have very different characteristics of cDNA lengths that might be, or different complexities, having equal representation of all the species that you're concatenating is not something that we've tested. That's the only thing I can say about this. Let's see. So here's a question for David. Have you tried using salmon with long reads?
Yes, I have tried also running salmon with long reads. So I mean, salmon has these two modes, right? This basically quasi-mapping mode and basically an alignment mode where you provide transcriptome alignments. And so for long reads, yeah, the quasi-mapping the k-MER-based approach doesn't really work or make sense for long reads. So you're forced to use this alignment approach. I have used this. One thing is that there is an error. So salmon has these error models that basically try to model sort of error distributions and sort of improve quantification through that, to the best of my knowledge. And as far as I know, there is an error model for nanopore, so NT, but as far as I know, there's no error model for PacBio for salmon yet. So I have tried it without an error model, but obviously then when you run it without an error model, it won't work as well as with an error model. And kind of, I mean, I'm not showing it here, but I found oarfish to give better results than salmon, but that may also have been because I did not use the nanopore error model for salmon.
Gotcha. Let's answer one more question before we close the webinar. This is for you, David. So you mentioned long-read biases in regards to quantification. Have you worked with tools such as, I think, Lika, LIQA, or NanoCount, which attempts to account for these biases?
Yes, so Lika was exactly the tool that I was referring to. So they have this approach where they basically estimate the survival probability and try to account for truncate reads through that. I think this is a very good approach. NanoCount as well. So I have started running these, but so in particular, Lika was quite computationally expensive, and so that was the main reason we didn't include it here, because it was just even compared to some of the different runtimes Lika was just kind of infeasible to run, also because parallelization was very difficult. But yeah, I think that is where things will go, right? This is also what happened with short reads. People are modeling all kinds of different things, and in short reads, things like fragment length distribution and so on, right? And I think this will become more and more with long reads, whereas currently a lot of the methods are still rather naive, and I think this will change in the next years.
Absolutely. So if there are any remaining questions that we haven't answered, we usually follow up with them over email. My closing comment is thank you for coming to the webinar. Please fill out the 2-minute post-webinar survey. We really appreciate your feedback. And to keep up to date on our webinars and events, please visit pacb.com/events. Thank you again, and I'll see you next time.